Hello friends, we are again with our new video in which we are going to discuss about gluconeogenesis. In the last three videos we have discussed about the glycolysis, that is the breakdown of glucose as energy stored in the form of glucose. So we break down the glucose so that we can consume, our cell can consume the energy. After that, the TCA cycle, which is a Krebs cycle, or we can say citric acid cycle 2, as well as the pentose phosphate pathway. Now in today's video we will discuss about gluconeogenesis. What is gluconeogenesis, what are their precursors, and how the cycle run, as well as we will study about the regulation of glycolysis. How can we regulate the glycolysis? Okay, so gluconeogenesis. So it is the formation of new glucose, or I can say it is the synthesis of glucose. As gluco means glucose, neo means new, and genesis means the synthesis. So gluconeogenesis is the formation of new glucose molecules with the help of non-carbohydrates. What are the non-carbohydrates? That is the lipid, the proteins, okay, etc. Now, so it is a universal pathway, that is, it is found in all animal, fungi, plants, as well as in microorganisms. In higher animals, it occurs in the liver, whereas in smaller extent, it occurs in the kidney cortex. Okay, and here are the precursors of gluconeogenesis pathway. That is, first is the glycolytic product like lactate, pyruvate, and glycerol. And second is the citric acid cycle intermediates. The intermediate of citric acid cycle is the isocitrate and cis-aconitase, etc. So those were the intermediates of the citric acid cycle. And the last that is that is amino acid, of which are known as gluconogenic amino acids, that is lysine and leucine. It is clear that it does not work as a substrate in this pathway. It helps to produce acetyl coenzyme A when it gets degraded. So now we will see how can we produce the glucose, how we get the glucose, as the remaining glucose will break down to get the energy. Now how extra glucose we will get from these cycles. So the first we get our lipid in the form of glycerol. As it is a glycerol, so we can break down glycerol to get the glucose. So we can get the glucose from lipid, and second is from protein, which are precursor to, that is glucogenic amino acids. What are the glucogenic amino acids? Yeah, that is lysine and leucine, which break down into acetyl coenzyme. So that is a glucogenic amino acid. Now the third we get from lactate. You guys know that milk contain the lactose. Okay, so it is stored in the form of lactate. So we can utilize lactate too to get the new glucose. That is by using their alcohol. And alcohol means the OH group. Okay, and the last, not the least, from pyruvate, their ketone group. So from these four we can generate our new glucose. Okay, so before we proceed, I must acknowledge that the process only occurs when we overeat. Okay, this process only occurs when we overeat, as well as uh, now we will see how lipid, that means how glycerol gets converted into glucose, how we get glucose from the glycerol. Here you can see glycerol first gets converted into glycerol 3-phosphate. After that it gets converted into dihydroxyacetone phosphate. Maybe you remember guys, the dihydroxyacetone phosphate occurs in the glycolysis pathway when glyceraldehyde 3-phosphate gets converted into 2 cycles. First it forms dihydroxyacetone phosphate, and the next it forms 1,3-bis phosphoglycerate. So here this glycerol gets converted into glycerol 3-phosphate, and afterwards dihydroxyacetone, and the after that glyceraldehyde 3-phosphate forms, and the reverse glycolysis cycle begins, due to which we get the last product, we get the glucose. And now we'll see the second. Here from glucogenic amino acid, that is another example is also alanine. Here we use alanine, which gets transaminate and forms pyruvate plus alpha ketoglutarate. Okay, what is the transamination? It is the formation of amino acid with the help of other amino acid by its alpha amino group. Okay, when we add alpha amino group to the amino acid alanine, then it converts it into pyruvate plus alpha ketoglutarate. And I hope you remember pyruvate is the last product in the glycolysis cycle, as well as alpha ketoglutarate is the one of the product in citric acid cycle. Therefore, both the cycle gets reversed. And the third, here lactate gets converted into pyruvate when CO2 gets added to it. Carboxylation occurs. Carboxylation is the addition of CO2. So when carboxylation occurs, lactate gets converted into pyruvate. Here NAD plus gets converted into NADH plus H plus. And this when process occurs in anaerobic condition. In anaerobic condition, lactate gets converted into pyruvate, and rest glycolysis, reverse of glycolysis cycle. And at last, so in our previous two precursors, the glucogenic amino acid as well as in lactate, we get pyruvate. Okay, pyruvate. Now I will tell you how pyruvates get converted into glucose, because therefore there are also irreversible cycle involves too in the glycolysis. So here you can see pyruvate first gets converted into oxaloacetate in the help of carboxylase and biotin. Okay, biotin is the precursor of bicarbonate, that is HCO3. So it is a precursor of bicarbonate, which is found in cytosol. So this pyruvate, when gets converted into oxaloacetate, then it is the reverse of Krebs cycle, where oxaloacetate gets converted into malate. And after malate, after which decarboxylation occur, where is the removal of CO2, and GTP gets converted into GDP with the help of PEP enzyme or carboxycarbokinase enzyme. It forms phosphoenol pyruvate. If you remember glycolysis, when phosphoenol pyruvate gets converted into pyruvate, it was an irreversible cycle. It was an irreversible cycle where pyruvate can't get converted into phosphoenol pyruvate back. So here, to convert pyruvate to phosphoenol pyruvate, here the oxaloacetate and malate are used. So here malate gets easily converted into phosphoenol pyruvate, and GTP gets converted into GDP. Due to removal of the CO2, the structure of ATP gets converted. Therefore, here we use GDP in the place of ADP. Okay, so from up to this cycle, up to this cycle from pyruvate to malate, it occurs in the cell cytosol. Afterward, the phosphoenol pyruvate gets a reverse procedure of glycolysis, fructose 1,6 biphosphate form, and fructose 1,6 bisphosphate is the irreversible process. Therefore, fructose 1,6 phosphatase are used to convert fructose 1,6 bisphosphate to fructose 6-phosphate, and at the last we get glucose with the help of enzyme phosphatase. Okay, from phosphoenol pyruvate to glucose, it occurs in mitochondria. Hope you understand easily how the pyruvate gets converted into a last glucose. Here we learn how 4 precursors are used. First is glycerol, the glyceraldehyde and dihydroxyacetone, and the rest is glyceraldehyde 3-phosphate. Second one was the glucogenic amino acid, that is the aldine. When the alpha amino group are added to aldine, it converts into pyruvate plus alpha ketoglutarate. And the pyruvate, how pyruvate gets converted into glycerol, it's glucose, it's in front of you, the cycle. And the last one was the lactate. Again, it's converted in, it gets carboxylation occur and pyruvate forms, and this cycle runs. So here, with the help of four precursors, our new glucose molecule occurs. Okay. Now, at last, we will see how we regulate the glycolysis cycle. So this is our glycolysis cycle. How we regulate the glycolysis cycle, we will learn over here, because we require a cycle. It is very important, because the enzymes which help to break glucose into pyruvate, those enzymes are responsible for the regulation of this cycle. Now, here is the three main enzymes over here, which work as the activator as well as the inhibitor too. Okay, so there is first hexokinase. Okay, there is a hexokinase. Second, there is a phosphofructokinase. Okay, you can see here is irreversible process. Hexokinase are used. It is used for regulating the glycolysis. Irreversible process for phosphofructokinase, used for regulating the glycolysis. And the last is pyruvate kinase, also a irreversible process, help to regulate the glycolysis cycle. So the rate at which the glycolytic pathway operates is controlled primarily by these three allosteric regulation enzymes. Okay, because the reaction catalyzed by these enzymes are irreversible, a high AMP concentration activates phosphofructokinase and pyruvate kinase, and a high ATP concentration inhibits both the enzymes. Okay, here you can see the three enzymes, how they inhibit and what they inhibit. Okay, so hexokinase inhibit glucose 6-phosphate, so the cycle will end in second phase only. Okay, and if cycle not inhibit over there, then the second is useful enzyme is phosphofructokinase. It inhibits the formation of citrate and ATP. If the citrate and ATP doesn't form, Krebs cycle won't run. Okay, and it activates the formation of fructose 1,6-bisphosphate, fructose 6-phosphate, as well as AMP, adenomonophosphate. It activates these enzymes, and which inhibits citric and ATP. Okay, now pyruvate kinase. Pyruvate kinase inhibits the formation of acetyl coenzyme A and ATP. Again, if acetyl coenzyme A won't form, the Krebs cycle won't run. Then there is a inhibition of glycolysis with the help of fructose 1,6-bisphosphate and AMP. These are the activator of this enzyme. Okay, I must clear you that when in aerobic condition, pyruvate gets converted into acetyl coenzyme A, whereas in anaerobic condition it forms lactate or lactose, and at last it forms alcohol. So this is the regulation of glycolysis cycle. These are the enzymes involved in irreversible processes, okay, through which we control the glycolysis. And we also learn the precursors of gluconeogenesis as the formation of glucose. Okay, so here our all metabolic pathways completed. Okay, in next module we will learn about some of the carbohydrates. as well as the amino acids. Okay. Um, if you have any query, you can comment over this under this video. I'll give the link of our recipe metabolic pathway video too. Also, I must like to remind you, if you haven't subscribed or subscribe our channel yet, so you may subscribe it to get our new update, as well as press the bell icon too. Thank you. Rest on my side.